Hey there, science fans. Have you ever wondered what makes up the tiniest things around us? Like, what does a human hair really look like up close, or the wing of a fly? Get ready to explore a hidden universe with the amazing compound light microscope. This incredible tool is like a superpower for your eyes, letting you see things you never thought possible. We're talking about magnifying objects hundreds or even thousands of times their actual size. From studying cells to analyzing crystals, the compound light microscope is a game changer in science. So, put on your lab coats and let's dive in. The compound light microscope may seem a little intimidating at first, but don't worry. Once you understand the basic parts and how they work together, you'll be navigating the microscopic world like a pro. In this quick guide, we'll break down the main components of this amazing tool, making it easy for you to grasp the basics and get started on your own microscopic adventures. Let's start with the stars of the show, the optical components. These are the parts responsible for capturing and magnifying the light that allows us to see those tiny details. First up, we have the eyepiece or ocular lens. This is the lens you look through, and it provides the final magnification of the image. Think of it as a magnifying glass for the image created by the objective lenses. Next, we have the objective lenses, the real heavy lifters when it comes to magnification. These lenses are mounted on a rotating nosepiece, allowing you to choose different magnification levels. Each objective lens has a different magnification power, typically ranging from 4 times to 100 times. When you combine the magnification of the eyepiece and the objective lens, you get the total magnification. For example, using a 10 times eyepiece and a 40 times objective lens gives you a whopping 400 times magnification. Finally, we have the body tube, a simple but crucial component. This tube connects the eyepiece to the objective lenses, ensuring that the magnified image travels in a straight line to your eye. Without the body tube, the light rays would scatter and you wouldn't see a clear image. It's all about that precise alignment, folks. Now, you can't see anything through a microscope without light. That's where the illumination components come in. Think of them as the stage crew, setting the scene for the microscopic world to shine. At the base of most microscopes, you'll find the light source, typically a small but powerful LED bulb. This light source provides the illumination needed to view your specimen. Above the light source sits the condenser. This clever component gathers and focuses the light from the source, directing it upwards towards the specimen. A well-adjusted condenser is key to getting a bright and clear image. You can think of it as a funnel for light, ensuring that enough of it reaches the tiny object you're trying to observe. Last but not least, we have the diaphragm, usually located just below the condenser. The diaphragm acts like the pupil of the eye, controlling the amount of light passing through to the specimen. By adjusting the diaphragm, you can fine-tune the contrast and resolution of the image. It's all about finding the perfect balance of light for your specimen. Section 4. Bringing the microscopic world into focus. So we've got our light and our lenses. Now, how do we actually bring the microscopic world into sharp focus? That's where the adjustment mechanisms come in. These are the knobs and controls that help you fine-tune your view. On the side of the microscope, you'll find 2 knobs, the coarse focus knob and the fine focus knob. The coarse focus knob is your go-to for getting the specimen roughly in focus. It moves the stage up and down in larger increments. Once you have a general focus, the fine focus knob comes into play. This knob allows for incredibly precise adjustments, bringing the tiniest details into sharp relief. It's like fine-tuning a radio dial to get the clearest signal. Remember the revolving nose piece that holds the objective lenses? Well, that's also an adjustment mechanism. By rotating the nose piece, you can easily switch between different objective lenses and magnifications. This allows you to zoom in and out on your specimen, exploring different levels of detail. Lastly, we have the stage control knobs, usually located below the stage. These handy knobs allow you to move the slide, holding your specimen left, right, forward, and backward. It's like having a tiny joystick for your microscopic explorations. We're almost at the end of our microscopic tour. But before we wrap up, let's give a shout out to the supporting cast. the essential components that provide a stable platform for our observations. First up, we have the stage itself. This is the platform where you place your slide, typically held securely in place by stage clips. The arm of the microscope is another important structural component. It connects the body tube to the base, providing stability and acting as a handle for carrying the microscope. Always carry your microscope by the arm and base to avoid any accidental drops. And finally, we have the base. This sturdy foundation provides stability and houses the light source. It's the unsung hero of the microscope, ensuring that everything stays put while you're busy exploring the microscopic world. And there you have it, a crash course on the compound light microscope. With a basic understanding of these components and their functions, you're well on your way to unlocking the secrets of the invisible world.